Hello students, in this video we are going to learn about tissue systems in the flowering plants. Well, I've chosen a topic from anatomy of the flowering plants, that is your chapter number 6. So we begin with three types of tissue system in the plants: the epidermal tissue system, the ground tissue system and the vascular tissue system. Now beta, the epidermal tissue system is made of three components. First one being the epidermal cells. There is mostly a single layer of epidermal cells which are very tightly packed together. So they form a ring-like structure, and then these epidermal cells, they may have certain appendages over them. The first one being the root hairs. If it is going to be a root, then it is supposed to have some external protrusions of the epidermis. So they are just the outgrowths of the cytoplasm and we call them root hairs. They are always unicellular. So you need to remember this thing. Whereas trichomes, on the other hand, they are multicellular. Trichomes you will find on the stem or the leaves. You might recognize actually whenever you go to certain mountains or go for trekking and try to hold on to some grasses, then it actually makes you feel irritated or itchy sometimes. So that is due to the presence of trichomes, which are mostly present on the monocot plants. Fine. So in the monocot grasses, these are actually present over there to prevent the loss of transpiration. Now the next one, we'll be talking about vascular tissue system. Why we didn't talk about the ground tissue system first of all? Later on throughout the whole session, the ground tissue system is going to be discussed. So let us have a look at the vascular tissue system first of all. Well, the two permanent tissues, the two complex permanent tissues, that is xylem and phloem. These two tissues, they combine together and then they are going to form the vascular tissue systems. There can be three important types of vascular tissue system. Depending on the presence or absence of cambium, we can have two types of them: the open and the closed vascular bundles. Well, in the open vascular bundles, as you can see in the picture given below, there is a specialized tissue. So it is cambium which is present in between xylem and phloem. So if this is xylem and this is phloem, these both are actually separated by a very fine layer of special tissue, and this is called the so this one is called the cambium. So we need to remember cambium is a special layer that is going to separate phloem from the xylem. Fine. So coming to the next type of vascular bundles, well, we have the closed vascular bundles as in case of monocot plants. Now, how do we need to remember this thing, that this is found in monocots? Students, just see that monocots, they will not show the secondary growth. So, if secondary growth is absent, they don't need to become very thick. That means cambium will be absent. So, they do not have an intrafascicular cambium that is formed between phloem and xylems. They are simply attached together. They are present on one single axis. That's why we call them closed conjoint vascular bundles. Conjoint, you can remember it from the twins. Sometimes the twins, they are joined together by their head or by any other kind of a body part, and then we call them conjoint twins. So in this case, the monocot stems, they are going to have closed conjoint vascular bundles. The third type of vascular bundle is called a radial vascular bundle. Now what do we mean by a radial vascular bundle? This one is actually present in the roots. Alright, now in both the types of roots, whether the monocots or the dicots, we have an alternate arrangement of xylem and phloem. Actually, the radii along which the xylem and phloem are present, they both are going to be different in these cases. That's why due to having a different radii, the arrangement becomes radial. And where is it present? In the roots of monocots, that is the grasses, as well as the roots of dicot plants. So you are going to see this kind of alternate arrangement of xylem and phloem. Now students, there is an important term to be discussed, that is stele. So once we get inside the cortex, the innermost layer of cortex is called endodermis, and just inside the endodermis, whatever else is present, that constitutes the steel. So what is steel made up of? Number one, pericycle. Secondly, your vascular bundles, and then the pith. These three things largely constitute the steel. It's an important question already been asked in the NEET exams. Moving further, we come to the ground tissue system. Now the ground tissue system is something that is present in between the epidermal and the vascular tissue system. Well, what makes the ground tissue system? All the simple permanent tissues, the parenchyma, collenchyma and sclerenchyma, these simple permanent tissues, they are going to form the ground tissue system, right? Now parenchyma cells, they are usually present in the cortex, the pericycle, the pith and the medullary rays. Just remember, medullary rays are the ones that are going to form the cambium. So they give rise to the cambium. So these are parenchyma cells. Parenchyma cells, they are already differentiated. That means they will be de-differentiated. They will start dividing once again. They become secondary meristem. Then they are going to form the secondary meristematic tissues. So, in the ground tissue system, first of all, we will be discussing the ground tissue system of a leaf. Well, students, in case of leaf, we do not have a cortex. The ground tissue system is called mesophyll in the leaves. That means something that is present between the upper and the lower epidermis. Alright, now this mesophyll, in case of dicot plants, we can divide it into two. This is in case of dicot plants that we can divide the middle tissue, the mesophyll tissue, into two types, the palisade mesophyll and the spongy mesophyll. We will be looking further in the diagrams that palisade mesophyll is given this name all because it has very tightly packed cells. It contains a large amount of chloroplast and hence it is going to carry out the process of photosynthesis. On the other hand, spongy mesophyll, it is also made of parenchyma cells having a little less number of chloroplast. Now these spongy mesophills, they are very loosely packed. Now when they are loosely packed, they will be having lot of spaces in between them, and these spaces then later on they are going to find out or constitute the substomatal space through which the water keeps on diffusing in between the leaf cells for transpiration purposes. Now students, it is important to remember it is because of this arrangement of palisade and spongy mesophyll that the leaf of a dicot will be having reticulate venation. So if you remember the reticulate venation, like this network of veins is present. This is just because they are going to have two different divisions of the mesophyll tissue. Whereas in case of monocot plants, we do not have the division of mesophyll into palisade and spongy, and thereby we have parallel venation of leaves. So the monocot plants, they are the ones that have parallel venation, whereas dicots are the ones that are having reticulate venation of the leaves. Now the next point, epidermis is single layered, whether it is the upper epidermis or the lower epidermis. Upper epidermis is called adaxial, whereas the lower one is called abaxial. Just remember the words ad, upper side, ab, lower side. Often the leaves, they are covered with a waxy covering which is called the cuticle. You people know the reason. Cuticle is going to prevent the excessive loss of water. It is present in the leaves, absent in the roots. Otherwise, roots will not be able to absorb the water, right? Now, the last important thing, something extra which is present only in the leaves, but that is the stomata. Now, what is stomata? Stomata, they are some tiny pores that are present on the surfaces of the leaves, especially on the lower epidermis. Now, when I talk about the lower epidermis, then again I am talking about the dicot leaves which are dorsiventral. Dorsiventral means they are going to have dorsal and ventral surfaces completely separate. But in case of monocots like the grasses, we have isobilateral leaves where the number of stomatas is going to be same on both the sides. Dicot plants will be having more number of stomatas on the abaxial side, that is the lower epidermis. Fine. Whereas lesser number or fewer number of stomatas will be present on the upper epidermis. Okay, one point I missed out. The type of stomata, it also varies. If I am talking about the dicot plants, the dicots, they have different kind of stomata. In the diagram, you can see the stomata, they are enclosed by guard cells. But what is the shape of guard cells? They are bean shaped or kidney shaped. Whereas in case of the monocot plants, the stomata, they are dumbbell shaped. Not stomata, but they are guard cells. But the guard cells, they become dumbbell shaped in case of monocot plants. So this is going to form a basic difference between the monocot leaf and the dicot leaf. Coming to the next slide, this slide shows you the gross anatomy of a dicot leaf. Well, in this particular slide, we can see the adaxial and the abaxial epidermis, that is the upper and the lower one, and the more number of stomatas, they are present on the lower epidermis, right? Now, this space just above the stomata, these spaces, they are called the substomatal spaces through which water is going to transpirate. Fine. And this is the palisade parenchyma. So palisade parenchyma is just arranged in a parallel manner, like one tissue in the front and the other one at the back. So this is at the back. Now since all these are tightly packed, we will call them palisade. Whereas the spongy, you know what is a sponge like. Sponge has lot of air spaces. That's why the name spongy mesophyll has been given. It is because it encloses a lot of air cavities. That's why we say that in a dicot leaf, the mesophyll tissue, the whole of this entire middle part is called the mesophyll. It is divided into two regions. Now, this is the vascular bundle that is enclosing xylem and the phloem. These vascular bundles, they are present only along the veins. This is something you need to remember. All right. So coming to the next slide, the monocot leaf. It's quite a clear one. I have not labeled the slide for you people. You can label it later on. So what are we going to see? Have a look. On the very top side, the top epidermis, that is the adaxial epidermis, while the lower one, this is the abaxial. Just remember the word ad and ab. Then this A part, it represents the xylem. This B part represents the mesophyll tissues. You can clearly see there is no differentiation between palisade and spongy mesophyll in this particular case. So, where is the phloem in this case? This D part that is represented here, but this all is the phloem part. Now, we need to see that there is no cambium, hence the vascular bundles, they are of closed type. They are conjoined and they are closed. Now, beta, in this slide, some of the cells, they often become very large. On the epidermis, some cells, they become very large and we call them bulliform cells. We call them bulliform cells. What is the task? What is the task of these bulliform cells? Well, these bulliform cells, they are going to absorb a lot of water. So when they can hold more amount of water, that means they are going to keep the leaves turgid and they are present only in the monocot leaf. So by the end of the slide, you will be able to differentiate many times between the monocot and the dicot leaves. All right. So bulliform cells, they help to absorb a lot of water, keep them turgid, and if there is a shortage of water, they will help the leaves to roll their surface, thereby preventing the loss of water by transpiration. And here you can see if one stomata they have shown on the upper epidermis, one stomata on the lower epidermis. That means the number of stomatas is going to be same. So that's why we call it an isobilateral leaf. Now, this is all about the stomatal apparatus. The stomatal apparatus. Beta, what is apparatus? Something that is needed to carry out an experiment. Well, the stomata, they are the tiny pores which are guarded by the guard cells. Guard cells, they carry chloroplast in them. You must remember guard cells are the only epidermal cells that are having chloroplast and that can do the process of photosynthesis. So what actually forms the stomatal apparatus? It is the stoma, that is the aperture, the pore. Then the guard cells, two guard cells which are bean shaped in case of dicot and which are dumbbell shaped in case of monocot. Then some surrounding subsidiary cells. When they come up all together, they are going to form the stomatal apparatus. Now, we start with the gross anatomy of the stem. So this is a hand-drawn diagram. You need to draw these type of diagrams for your exams also. Keep on practicing these diagrams again and again by drawing. Well, we can notice very clearly this outermost layer, beta, this outermost layer, that is the epidermis. Now in the stems, you can see small hairy extensions. Now what are these hair-like extensions? Of course, they can't be the root hairs when it is a stem. So they should be trichomes. Just remember, these hair-like extensions, they are trichomes, and they are the outer appendages of epidermis. They are a part of epidermis. Now on the outer surface, sometimes cuticle can be present. If the stem is herbaceous, it is having a thin waxy coating of cuticle. Then inner to the epidermis, we have the second tissue, that is the hypodermis. You should not be confused. Of course, this is going to be a part of cortex. Cortex is formed by the combination of hypodermis. So first of all, on the outer extensions of the cortex, we are having the hypodermis. Now, but a hypodermis is made of collenchyma cells in this case. Collenchyma cells. Whereas in case of monocot stems, you will see that the hypodermis is there, but it is made up of sclerenchyma cells. What could be the reason behind that? See, dicot stems, they are already having the process of secondary growth, which is going to provide hardening to them. So collenchyma will provide some flexibility to the plant tissues. But monocots, they are already so flexible. They need a little bit of more mechanical support, more mechanical strength. That's why they are going to have sclerenchyma. Moreover, they do not have secondary growth. So remember, in monocots, the hypodermis is made of sclerenchyma, which is going to provide a bit hardening effect to them. But in this case, the dicot stem, hypodermis is essentially made of collenchyma cells, which is going to provide elasticity to the organs. Right. Now, just inner to the hypodermis, which is made of a few layers of collenchyma, we have the cortex. Now, what is the cortex made up of? This cortex is entirely made up of the parenchyma cells. Now, the parenchyma has the basic function to carry out either the photosynthesis. If it is a green stem, a herbaceous stem, it can carry out the process of photosynthesis. But most of the times, parenchyma cells, they are going to store food in them. Then the innermost tissue of the cortex, this wavy layer that is very visible, this wavy layer, this is the endodermis. That is the endodermis. So just remember, what is the endodermis? That is again made of one single layer of cells. One single layer of cells that is going to form the endodermis, and these cells, they are very tightly packed. That means they are essentially not going to have any kind of space in between them. And hence, we say that endodermis is made of single layer of tightly packed cells. Now, this endodermis is going to store a lot of starch. So, we also call it starch sheath. What do we else call it? The starch sheath. In case of the stems, of course, in case of the dicot roots, you will see endodermis is going to be covered and surrounded with a Casparian strip. So, few differences are there. Notice them very clearly and the whole process becomes very easy. So, what is the shape of the vascular bundles? If you look particularly, it is a wedge-shaped vascular bundle. Wedge-shaped vascular bundle like this. Fine. So, on the inner side, you are going to have the xylem. On the outer side, the phloem is there. But you can clearly see that the xylem and the phloem, they are separated by cambium. Fine. That means secondary growth will be present. Just remember the name of the cambium, that is intrafascicular cambium. So, in between the xylem and the phloem bundles, what else is present? So, let us notice that. Here we can see clearly that some cells are present here on this axis. But these are going to form the medullary rays. Of course, they are going to be formed of parenchyma cells, but we call them medullary rays. Then this interior part, that is called the pith. Fine. So, all this pith, essentially the cortex and the medullary rays, what are they made up of? They are made of parenchyma cells. This is what we have studied earlier as well. In the next slide, good, we see a colored beautiful diagram of absolutely the previous one. This is again a dicot stem. So if we ask you to draw a dicot stem, you can show the sunflower or the mango or any of the other plant, but remembering the examples is very important. Remember sunflower, mustard, all these things, they are going to be dicots. But if I say maize plant, students, maize is going to be a monocot. Sorghum is going to be a monocot plant. So you cannot show this kind of a diagram. Fine. So we can see the cortex. What is the cortex made up of? By the combination of hypodermis on the outer side, and remember HC. Remember HC Verma sir of physics, his famous books on physics. Do you remember that? So remember hypodermis of the dicot stems, it is made of collenchyma cells, followed by the general cortex, that is the parenchyma cells, and the innermost layer, that is the endodermis. And what is the endodermis of a dicot stem called? It is called the starch sheath. It is going to be storing lot of starch. And this interior part, that is the pith. Fine. So where are the medullary rays? You cannot see in this diagram. Let us mark them here. The parenchyma cells that are going to pass out in between, they are called the medullary rays. And what is the function of medullary rays? Later on, they are going to form a cambium, so that it can form a complete ring of cambium, and a complete vascular cambium is formed. Next, moving to the next slide. Alright. Now students, this is the TS of a monocot stem. So monocot stem, it's a beautiful one. Pith is absent in this case. There is no pith. This entire part, this is the ground tissue. This entire part is going to be the ground tissue. You can see this is entirely filled with parenchyma cells. What kind of vascular bundles? Earlier we saw that vascular bundles, they were of ring type. They were all arranged in the form of a ring. But in now the present slide, that is the monocot stems, we can see that vascular bundles, they are all scattered. They are all scattered within the ground tissue. So the ground tissue is again made up of cortex, or the cortex, that is all one and the same thing. What is going to be the outermost layer? The epidermis, followed by the hypodermis. But what is the hypodermis made? In this case, it is going to be made of the sclerenchyma cells. Just remember sclerenchyma because they are dead cells. All the sclerenchyma having lot of thickenings of the lignin. So they are going to provide somewhat hardness to this particular stem. Vascular bundles, although they are scattered, but they show some special type of effect. Well, on the outer edges, on the outer periphery, we can see they are smaller ones, small vascular bundles on the periphery, whereas larger ones are present more towards the center. So this is how you are going to explain the tissue of a monocot stem. That's it. Coming to the next slide. Well, this is again presenting all the same facts. You can simply try to have an overview. What is going to be the arrangement of xylem? You can see the xylem is going to make either a V shape or a Y shaped fashion over here. But phloem, in this case, students, phloem is made up of sieve cells, sieve tubes, companion cells, they are present. Phloem fibers are present, but phloem parenchyma is going to be absent in the monocot stems. So this is just one exception that we need to keep in our mind. Now, this is a dicot stem. Simple practice slide, it is there for you people. The outermost layer, that is the epidermis. Then the inner one, that is going to be the hypodermis, followed by the cortex. This one single layer is called the endodermis, right? Epidermis and the endodermis. So just keep on revising all these things for yourself and try to sort it out. Now, if the name was not given, how you could have recognized that this is a stem or a root? Well, the answer is very simple. You just need to look here. This is the cambium, and cambium will be present only in case of dicots. Fine. Then how do we know that this is a stem? By looking at the vascular bundles, we can clearly judge that the vascular bundles, they are not radial. We have radial vascular bundles in the root. Okay, so now we come to the monocot and the dicot root, which is the last part of the chapter. Now, in the beginning, a small little hand-drawn diagram has been given. Let us study it in the form of a flow chart. Why I drew this flow chart? Just to give you an idea that you can all remember all these things in this particular manner. So, when we move from the outermost layer, beta, this is the epidermis. Epidermis is made of one single layer of cells. What are the appendages? Root hairs. They are unicellular. Remember trichomes? They were multicellular. Then the next tissue, this is the hypodermis. These cells, they are going to form the hypodermis. Hypodermis may be present, it may not be present. Then the innermost part, next one, just below the hypodermis, that is the cortex. That is the cortex. And you all know now, hypodermis, cortex and endodermis, they are going to form the ground tissue system. Ground tissue system. Pericycle, pith, xylem and phloem are going to form the vascular tissue system, whereas the epidermis and the root hairs, they are going to form the epidermal tissue system. So this is a handwritten note for you people. And what is a stele? For just remembering, it is a combination of pericycle, pith, xylem and phloem. Let us have a broad overview of this slide. Right. Now, since it is a dicot root, students, okay, how it is a dicot root, how ma'am comes to know about that? First of all, you can see a lot of root hairs which are present. Very few root hairs will be present in the monocots. Secondly, you can see the Casparian strip. Where is the Casparian strip? In the endodermis. So this layer, it forms the endodermis, and inside this red colored tissue, beta, this is going to form the Casparian strip. Now, what is Casparian strip? It is something different from the cuticle. It is going to prevent the entry of any waste substance, unwanted minerals into the xylem and phloem. So, it is a big checkpoint. Where is it present? In the roots, because they are the ones who are going to absorb water from the soil, right? So, Casparian strip is made of suberin. Remember, it is made of suberin. And cuticle was made of cutin. Alright. So the inner layers, okay, the cortex is made up of parenchyma cells. Cortex, which is made of parenchyma cells. How do I remember that they are parenchyma cells? But intercellular spaces, you can see clearly they are present. So these intercellular spaces, they are present only in the parenchyma cells. Now the next one, endodermis, then inner to the endodermis we have a pericycle. Fine, then what is there inside the pericycle? We can see the vascular bundles that are present. Very small pith will be present at the center, very small pith in case of the dicot roots, and the xylem is arranged in the form of a plus-like manner. You can remember it in the form of a star that I told you in the class, right? So this is going to be the xylem. So where are the phloem parts? The phloem is present as patches in between the xylem, like this. These are all the phloem tissues. So which arrangement is this? That represents radial arrangement of vascular bundles. Now it has become really simple. So now you need to guess what is this slide actually representing. The first thing that I can notice is the vascular bundles. The vascular bundles, they are radial vascular bundles. That means it should be a root. Now the second thing, whether it is a dicot root or a monocot root. So you can see there is a big pith in the center, and there are lot of vascular bundles. It is polyarc xylem. The xylem is polyarc. So if so many xylem vessels are present, it should be a monocot root. Remember this thing. It should be a monocot root. Alright. So this is the most basic criteria that you are going to look for. If it is radial vascular bundle, it should be root. Then if we are having very few number of xylem, see, we are coming back to the back slide. This is hardly 4 xylem they were present. We can call it tetrarch xylem. Tetrarch xylem. It can be either 3 or 4. That means few number of xylem tissues are present. So this is going to be a dicot stem for sure. Fine. But if we have many number of xylem, polyarc xylem, it should be a monocot root. Then the next one, yeah, this is the entire same diagram, very similar but with the complete labeling. Now in this outer part, I can say with confidence, this is root here. This is not a trichome because it's a root. It's a root. The outer layer is called the epidermis. This pink layer that is shown here, this is the epidermis, and there is no cuticle. Then inner to the epidermis, here cortex is present, which is made of lot of parenchyma cells. Cortex which is made of parenchyma cells. Then the innermost layer, that is of cortex, innermost layer of cortex, that is endodermis, which is having tightly packed cells. So what is present just next to the endodermis? That is pericycle. Now beta, in this case, pericycle is not going to form any kind of secondary tissue. Just remember that pericycle is not going to form any kind of secondary tissue. Moreover, xylem parenchyma is absent in the monocot roots. Why? Because there is no lateral conduction taking place in the monocot roots. So, pith is very large. It's a very conspicuous and prominent pith that is developed. Dicots have very small or inconspicuous pith. What about the vascular bundles? They have polyarc xylem and secondary growth is absent. So on the basis of this slide, you can easily differentiate between the monocot and the dicot root. Looking towards another diagram of dicot root, here in the center we have a very small pith that is present, a very small minute pith that is present. Fine. So the pith is not very conspicuous. The xylem bundles are just 4 in number. They are tetrarch. Very few in number means it is going to be dicot. It is going to be dicot. Then phloem is present in the form of patches between xylem, right? So where is endodermis? Endodermis is present just in the innermost part of the cortex. Innermost layer of cortex is endodermis, outermost is epidermis, and in case of the roots we call it epiblemma. Unicellular hairs or the root hairs are also present. So students, that's it. With this we end our today's bio module. Just keep on revising the slides again and again. Listen them as many times as possible so that the topic actually becomes very simple for you. All the best.